you, Mira. Thanks, everybody, for coming here, and I'm looking forward for a wonderful conference day. One thing that I thought I could say is that I forget his name. There was a guy, Amos Korchin. He was the head of neurology. Is he here, Amos? No? Okay. He was the head of neurology in Ichilov, anyway. And something that I heard in a meeting of the department that he said, when they wanted to nominate someone for something in the hospital, and someone in the committee said, it's not good because he is too young. So Amos said, don't worry, it's temporary, it will pass. So it will not remain like that. So I think it's true, of course, for all of us. And one additional thing that I could say before presenting the talk, always when I come to the conferences and they give me this, I say the one good thing about it is that I will know who I am. And in case I forgot, and there is a story about that on Peres, that the joke at least is on Peres, that he came to visit an old age home, and he gave a talk, whatever, and afterwards when he was walking in the corridor, he wanted to see if he is still recognized. So he stopped someone and says, do you know who I am? He said, no, but if you go to reception, they will tell you. Okay, so the first talk, always I enjoy, I like having Ilana in the first row because I can hear her laughing all the way. The first talk is by Professor George Weinstock, who is the Irwin Family Chair and Director of Microbial Genomics in the Jackson Laboratory of Genomic Medicine. And his talk will be about, do microorganisms have anything to do with aging?
We are good. All right, well, good morning, everybody. Thank you. I'm sure that this didn't add years to our lives or anything. We're okay. We're going to be fine. Do microorganisms have anything to do with aging? So we're going to talk about the microbiome and its possible role in aging. And I sort of approach this as kind of like a group meeting. It's not a lecture or anything. I'm just going to tell you about some of the projects that we're doing, where we are with it, what our thinking, what our motivation is, and stay tuned for the future because that's when we'll do more. But I'd like to wish you all a happy Pi Day. Now I know your calendar is different than my calendar, but on my calendar this is March the 14th, 3.14, so we call it Pi Day. So happy Pi Day, everybody. Okay, so since some of you may not be that familiar with the microbiome, let me start out by, do we have a pointer, a laser, or anything? You can use the mouse, yeah? Okay. Very brave to lend this to us all. Okay. Here we go. Okay, so microbiome was a term invented not that long ago, 2001, by Joshua Lederberg, a very famous microbiologist, the ecological communities of microorganisms that share our body. So he had this concept that we were a superorganism, that we not only had our human genome stuff and our human cells, but we also were colonized by many, many, many microbes that made many communities and were working synergistically with the human side of us. And so they're really all together we're a superorganism of all of these things. So there's a lot of ecological thinking that goes into thinking about the microbiome. And then another term that is often used is metagenome. So if you think of some part of our body, your mouth or something that has a community of microbes there are lots of different species. So you think of it like if it was a forest, there would be birds, there would be snakes, there would be plants, and everybody would have their own genome. together, that's the metagenome. And so often your metagenome is referred to as your second genome because it's providing all kinds of functional capabilities. All those genes have other pathways. They help you degrade food. They help you extract nutrients. They synthesize molecules that you don't synthesize for yourself. They do all kinds of wonderful things for you. And so that's your second genome. And together, you're a superorganism of all your microbes as well as yourself. So that's what we're going to talk about. And here's one of us, and you have 10 to the 13th cells in your body, or so they say. And if you counted the number of bacteria that were in your body, it's even larger. You have more bacteria, so you're mostly bacteria. You're all microbial communities sitting out there, and your body is just a framework, a niche for you to live in. And they have even more viruses, 10 to the 15th viruses. So we're going to talk about all of these today. But you're basically just loaded with microorganisms. And so the question is, what is that going to have to do with aging? It's got to have something to do with aging. It can't just be a parallel universe. It's really tightly integrated with everything going on in you. And if you think about genes, like I said, you have this second genome here, all of the genes from the microbiome, millions of those genes from all the organisms, and the human genome has of the order of 20,000 genes in it, very important genes, but right now your gene activity in your body is mainly the microbes cranking away, and you have a lot of human genome activity too, but the microbes are the dominant thing, doing all kinds of things. Okay. Here is what one of those microbial communities might look like. This is the saliva microbiome. So these are all different genera of bacteria, and the font is the relative abundance. You have a lot of Streptococcus and Prevotella in your saliva, and then there's lots of minor organisms, but they're not unimportant. They're just minor in terms of their abundance. And altogether, you have hundreds of different taxa of microbes. And the challenge in studying the microbiome is you take a sample, you take spit, you take stool, you take a skin, you take something like that, and you have a community, and it has hundreds of different taxa with a range of abundances, and you have to sample that and see what's there. The first question we always want to ask is, what's there? Who's there? And most of these organisms have never been cultured. Maybe they never can be cultured independently of each other because they're all dependent on each other in a community, or they're dependent on that particular niche in your body for certain nutrients or certain things. So they're difficult to grow. And I know of examples, we've done this, where you can only grow a particular bacteria if another bacteria is also present because it's feeding it with something special or something like that. So that's more of the challenge of studying the microbiome too. And then here are a number of different body sites. Each of these pie charts, the different colors are different genera of bacteria that are there, and you can see that different parts of your body have different communities, different amounts of those bacteria, although generally speaking, everything is everywhere. They're just in different amounts. And that's because some parts of your body have air, like your mouth, and some parts of the body don't have oxygen, like your gut, and some have a lot of nutrients and some don't, and some are dry and some are oily, and you have all those different communities. Just like in ecology, you have forests and you have lakes and you have tundras and you have all kinds of things. In each of those, you have different species of animals and plants that can live in those environments. Same thing in your body, but all at the microorganism scale. And this is the way that we study them. Here's a sample. This could be that saliva sample, or it could be a stool sample. One of our favorite things to look at is the 16S ribosomal RNA gene. This is the barcode for each species of bacteria. You'll hear me use the word operational taxonomic unit, or OTU. It's the operational definition for a species of bacteria, and it's just based on the sequence of the 16S ribosomal RNA gene. Why that gene? It's a gene that every bacteria has, and so if you sequence it, determine its DNA sequence in every bacteria, you'll have that barcode for everybody. And it's something that just changes very slowly, evolutionarily, one nucleotide change at a time. And so there's a nice evolutionary relationship between all of the organisms. And so it's a good thing to use as a barcode. There's not scrambling of it that goes on or funny things. So if we target that gene and we sequence that, we're taking the census of all the different types of bacteria that are present, the different taxa that are present, and then the number of sequences that we get for each taxa is its abundance. So here's a data point from a person's saliva, one person. Here's the different genera of bacteria, and here's the relative amounts to each other. And that's what we crank on when we do analysis. And then the other type of analysis we do is we just take all the nucleic acids and just sequence everything that's there, either the DNA or RNA, which we convert to cDNA. And this allows us to not just look at the bacteria, because we're not just looking at the 16S ribosomal RNA gene. We see all the viruses, fungi, yeast, protists, all the other things that are there, or we can just ignore the particular type of organism and just analyze all these sequences in terms of what genes are present and figure out the enzymes, the biochemical pathways, the genetic variation, things like that. So these are all completely different windows, completely different views on what the microbiome is on a particular community. And that's the data that we work with in order to try to figure things out. Okay, so I'm going to tell you about 3 different aging-related projects that we're doing that have to do with the microbiome. One of them is with George Cashel, some of the aging researchers, as opposed to aged researchers. Some of the aging researchers in the audience may know George. He's the director of the Center for Aging Research at the University of Connecticut Health Center, which is right next to where my Jackson Laboratory for Genomic Medicine Institute is. And he was doing a study of influenza vaccine. Influenza vaccine doesn't work as well on older people than it does on younger people. And so he was doing a study of young versus old, and so he had people coming in. And so we started to collect stool samples from them to also begin to look at the human microbiome in young versus old people. So we were piggybacking on his thing. You can see we only ended up with 23 subjects. This is because some of the elderly people didn't necessarily poop every day. And so they would have a scheduled clinic visit to have their blood tested for the vaccine study, but they hadn't gone to the bathroom yet, so they didn't have a sample to bring in, so they rescheduled their appointment. So we were starting to interfere with his project. So after 23 people, we said, enough already, we'll just analyze those and we'll try to design this experiment better the next time around. So we had 10 healthy young people, 10 healthy elderly people, and 3 frail elderly people, and we looked at their microbiomes. So one of the things that you look at when you look at a community of microbes is its diversity. How many different types of organisms are there, and is the distribution of those organisms uniform? So if you have the same number of everything, that's like very diverse. But if 99% of the cells are one type of organism and then the others are another one, that's not very diverse. So there's all kinds of ways that the ecologists have defined diversity, and what's shown here are 5 different ways that they've defined it. And what we look at here is saliva samples or stool samples. And let's just look at one of these measures. This is the total number of organisms that are present, different types of taxa that are present, species. And so here's a saliva sample, and green is young, and red is old. And so you can see in saliva, the young people have greater diversity, more different types of organisms than old people do. But it's not always true, because if you look at a stool sample, it's just the opposite. In the gut, older people have more types of species of bacteria than young people do. So, and this holds true if you look at all these different types of measures of diversity. So here's something right off the bat, that there is a difference in age. It depends on the particular part of your body, the particular microbial community you're looking at, and that okay, now we've got things that we now know when we dig more deeply into this to study it, we're going to do that. Now, diversity, just in general, there's a feeling that the more diverse a community is, the better it is, the healthier it is. You can think of it like a lake. A healthy lake has lots of different types of fish in it, but if it gets polluted, most of the fish die, and there's only a few types of fish that are there, and so the diversity of species of fish in a lake is a measure of how healthy it is. Well, it's not quite so simple for the humans, because here, obviously, in one case, if you're looking at saliva or you're looking at stool, the more diverse one could be young or the more diverse one could be old. So it's not going to be a simple relationship like that. It's going to have to do with other things as well. Okay, now, here's the thing that we would really like, and I would really like. Can we find the fountain of youth bacteria? You know, can we find that bacteria that young people have, and if you take it in a pill, it makes you young again, or, you know, makes you feel young, or something like that. So we're now looking at, in all of these different samples, are there bacteria that are specific for young people or specific for old people? And the answer is, yeah, it looks like we've got lots of candidates for that. These are just a few examples. So here we're just going to look at bacteria in stool samples, but the same things hold true in saliva. And this is a particular type of bacteria, a particular species of bacteria. Here's in young people and here's in old people, and you see many people don't have this bacteria, but if they have it, they're young. And old people, we just don't see it very much at all. So this is a young specific bacteria. And here are 3 examples. Each of these is a different species of bacteria, and you can see the old people have it and the young people hardly ever have it. So yeah, if you didn't know if you were old, you know, you could see, yeah. But I'm going to come to biomarkers for age in a little bit. It's an important question, absolutely. So, you know, where do we go with this? So aside from the fact that can we take this bacteria and give it to an elderly person and make them young again? Maybe we can. We'll see, because it's obviously depleted in elderly people. The other thing is, you'd like to, for all of the very specific clinical symptoms of aging, you know, problems with constipation or with your teeth or your gums or, you know, your skin or all these different things that are happening, can you tie those to particular types of bacteria? Are there a relationship for that? Because it's not necessarily going to be one bacteria is going to affect everything, but different ones for different communities will affect things. And so that's the logic here, is to try to reduce this down to particular microbiome effects and particular microbes that are causing those effects. All right, so now, since we had this great question about biomarkers, there's actually a program in the National Institutes of Aging called Development of Valid, Reliable Markers of Aging-Related Biological Mechanisms for Human Studies. So really what they want are markers, you know, molecular markers that have to do with different aspects of aging. Maybe they'd be a better diagnostic, maybe they'd give you some insight into the mechanisms of those particular aspects of aging, whatever it is, but we're part of that consortium, we're part of that program. What are the types of things that some of the labs are doing? There's one that looks at methylation of DNA, particular genes and the patterns of methylation that you see. Turns out this is a remarkably reliable clock. And it's because when you methylate something, that change is fairly stable. It's not like transcriptional patterns, which are also used for clocks and are interesting, but they're changing in shorter time frames. Methylation apparently is more stable, so it's an even better marker for that. And the clock is pretty amazing. They can relate it exactly to biological age based on the methylation pattern of a number of different genes and everything. But our project is to use the Virome. And you don't have to read all this. The basic concept here was, as you go through life, you get infections and you have viruses and your body clears out the viruses, but it doesn't completely get rid of all the viruses. And so just like you're loaded with bacteria, you're also loaded with viruses right now, even though you think you're okay, you're all infected. And so as age goes on, you accumulate those, that's our hypothesis, and those viruses stimulate inflammation and other host responses. And so that becomes kind of like a chronic inflammation. And so over time, as people age, they will develop consequences of that chronic inflammation and things. So we're using viruses, that hypothesis, the viruses or maybe particular types of viruses could be very useful markers. So it's another part of the microbiome. And so in our experiment, we look at all the viruses in people at different ages. We also look at all kinds of immunological markers, cytokines, other measures of inflammation and things, and we try to correlate viruses, cytokines, and clinical write-up of the particular individual and what's going on. So these are healthy young people. This was part of the Human Microbiome Project. And this is just looking at viruses in people. So here's 100 or so different people. And here's a bunch of different viruses. And you can see, like, here's a virus that almost everybody has. That's a particular herpes virus. Well known that everybody's got herpes virus. Then there's other viruses that just a few people have, and every kind of distribution in between. So if for all of these people, we now add up how many viruses did this person have and how many viruses did that person have, here's what it looks like. On average, this is in a stool sample, for example, average person has 4 or 5 different types of viruses that we can easily detect. But some people have 15 or 16 different types of viruses that they're walking around with, even though they're healthy young people. But it's this virome that we want to use as a biomarker that's related to aging. And we're just, and here's another important fact. How do we know that this isn't just some remnant of a viral infection you had and your system got rid of it and the virus is actually not doing anything? It's just some piece of DNA left over from that infection. And here's a kind of interesting piece of information. These are DNA preps where we look at detect the virus based on DNA. And so these are DNA viruses that's relevant to. You can see here RNA viruses, we don't see any DNA, that's a good thing. And retroviruses, which are RNA viruses, these integrate into the genome, so we do see some DNA from those. But of the DNA viruses, if we also now look in the RNA, you can see that many of these DNA viruses, we can see their sequences in the RNA, which means they're actively infecting, transcribing, replicating. Something's going on there. Okay. So here's like an early data point. Don't worry about all the different panels, just this is age. And this is number of viruses. And so you can see in a lot of these different ways that we're slicing and dicing this. As people get older, we do see more different types of viruses in them. So sort of that's the rationale for our hypothesis that maybe this is going to be an interesting marker as we dig deeper and deeper down into it. Okay, so the third aging project that we have, and this is sort of one of my favorites. This has to do with trying to understand how host genes control the microbiome. And the way you can think about it is, as you age, host gene expression will change, and those host genes that are being turned on or turned off are going to be related to age-related phenotypes like incontinence or something like that, something going on in your kidneys. And then down here, you could imagine as you age, it has a direct effect on the microbiome, whether it's those viruses or bacteria or anything. And it's actually that change in the microbiome that gives you the age-related phenotype. It's just directly the microbes are having something to do with it. And then there's this more complicated but more interesting, and that is that there are some host genes that are going to control the microbiome. And so as you age, you're going to change the host gene expression, and that's going to change the microbiome. And it's because of that change in the microbiome that you're going to have these phenotypes. So we're looking for all of those kinds of situations, and it's basically, for the cognoscenti in the audience, we're looking for QTLs, quantitative trait loci, genes in the host that control microbiome or some other phenotype relating to aging. So to do this, we use mice, And that's because we can use the awesome power of mouse genetics to do this. And I'll tell you in the next slide a little more detail. But this is a slide to show you that it's easy to find mouse genes controlling the microbiome. So what do we have here? These, on the x-axis that you can't read, these are the 20 mouse chromosomes. And this is a system called chromosome substitution strains, where we have one strain called C57 black 6. It has its 20 chromosome. It's an inbred strain of mouse. And then we have changed one of its chromosomes out for that chromosome from another mouse called AJ. And that will have a slightly different sequence on its chromosome. And so each of these 20 points here is C57 black 6, but it's got some genes from another mouse. Okay, so we're systematically varying that. And then these are those OTUs again. These are species of bacteria. So this particular species of bacteria, you can see its abundance changes depending on which chromosome from AJ we've introduced into black 6. So this just shows you that as you change a particular chromosome, you change the amount of this particular species of bacteria. In other words, there are plenty of loci in the mouse that are affecting that particular bacteria. And here are 4 other bacteria, and you can see that they all sort of change as you change those chromosomes. So there's a lot of genetic control by the host of the microbiome, and it's at the level, it can be at the level of individual species of bacteria. So this is very cool, and this is one of the things that we want to study. And here's the system that we use to do it. I'm going a little deeper into this just because of those of you in the audience who are interested in genetics. There's something called the collaborative cross, and the collaborative cross was done by taking 8 different founder strains, well-known strains of mice, as well as 3 strains that were wild strains to introduce a lot of variation, and crossing them with each other until you had a large number of hybrid mice, and then inbreeding those so that you ended up, they have about 100 of these now, where they each are inbred, and so they're homozygous, but they have different contributions from each of these founders. And so these are a new set of inbred strains. That's the collaborative cross strains, and then these collaborative cross strains are crossed with each other through another round to generate now a lot of very diverse things called diversity outbred mice. Think of diversity outbred mice as like the human race. They're sort of similar to each other, but everybody's different from everybody else also. And so every time you want to do an experiment that does this genetics, you generate a new cohort of diversity outbred mice, but they all come from the same source originally, and all of these genomes have been sequenced, so we can easily figure out exactly the sequence of each of those mice in that cohort. Okay. And here's another project we have about addiction, where we're trying to map host genes in the mouse that have to do with cocaine addiction. And this just shows you, here are the 20 chromosomes again. Here we're looking to see for a particular addiction phenotype, are there any regions in the mouse that show a particular mutation that affects addiction. And so the ones that have the high peaks are particular quantitative trait loci, places in the mouse where there is a gene that is affecting addiction. And here you can break, so this one on chromosome 12, we just have expanded that little region, and here are the 8 different founder strains, and you can see, we can say which founder strains are contributing to this addiction, the high ones and the low ones, and then here are the genes that are present there, and we can even identify there's a particular gene called DOC4 in this region. So this is the awesome power of mouse genetics. Here we're looking at a phenotype of addiction, and we can reduce it down to a particular thing. Okay, so that's what we're trying to do for the mice. So we now have mice that are 6 months, 12 months, 18-month-old, and we're running them through that same pattern right now. And this just shows you that just like with the humans, we can find particular bacteria that are specific for old mice or young mice in this system. Okay, I think I will just stop there. Here's the team that's doing everything, and you get the idea of what we do with humans and with mice in order to try to figure out what the microbes are doing and what relation they have with aging, and it has a lot to do with them. So thank you very much.
Questions, please.
I wonder why.
Because we're not studying it. I mean, there's a lot of things. Yes. What do you have in mind in particular?
In terms of aging.
Yes, I totally agree with you. I actually think that at some point we should be able to swing around and show how when we get down to the level of metabolism or growth rates or things like that, mitochondria are going to have a lot to do with the microbiome, because the mitochondria is an old bacteria, and and we can actually count mitochondria when we do our 16S sequencing, because we do pick up the mitochondrial genes for all of that. So we're aware of it, we just haven't jumped on it yet. Yeah. Jonathan?
Correlation.
Yes. Right. So let's take that last case with the mouse. Okay. So we find what we're trying to get at there is causation by showing here's a mouse gene that affects this particular bacteria, and then we can take a collaborative cross, an inbred strain of mice that has just that gene and just that mutation, and now we can correct it. We can do CRISPR genome editing and change the nucleotide and show that the phenotype changes. So that's one of the reasons that we go down that path, is it's an alternative way of really absolutely proving causation short of introducing the microbe in there. Now the earlier ones with the humans, where we just are seeing correlations. This particular bacteria is higher or lower or whatever in young versus old. Totally correlative. And what I was implying there is that, let's say one of those has to do with constipation or something like that. Now, the people who have that bacteria and they also have, this constipation phenotype, now how do we prove that? How do we now go from that correlation? And it's much harder with humans. But that's exactly the game that we have to play now to really prove there's something. National Institutes of Aging called these biomarkers because they're not sure how mechanistic we could actually get into in terms of proving causation. But the thing about a correlative marker is it's still useful as a diagnostic or still useful as a clock for aging or something, even if mechanistically you can't prove that it caused something. So it is an important aspect, yeah. No, these were human viruses. The same thing holds true with phages. We look at the phages, and they have a lot to do with affecting the microbiome by changing the amounts of the particular host bacteria that they have. That's like a whole other ecological predator-prey, blah, blah, blah kind of story. But these were human viruses. These were stool samples. Those were stool samples. But we see human viruses in just about any kind of sample that we look at. Yeah. Yes. It's too soon to tell, but that's the hope, is that they're not going to be like just two completely different stories that don't relate to each other. And in particular, one of the reasons for focusing on mouse genes and tying that to it is, at least with the mouse genes, we can now look at the cognate human gene and now have something to focus on in the human that gives us some mechanistic insight, maybe. So that's the hope, but it's too soon to say it's really one-to-one, yeah.
Okay, thank you.
Okay, thanks.